Hi everybody. So in the competitive classes, let's start the first year chapters. Let me begin with the chapter biological classification. Okay. So let me brief the chapter first, then we can move on to the questions. Okay. So just recall what are all the things you learnt in biological classification. Okay. So in this biological classification, you learned the classification of organisms. So classification of organisms means according to their characters, according to their similarities and dissimilarities, organisms are grouped into the categories. So those categories are nothing but classification. Okay. So under that classification, you are going to learn the systems of classifications. So with respect to all the organisms. Okay. Like a 2 kingdom classification, 3 kingdom, 4 kingdom, 5 kingdom and 6 kingdom classification. So under that you learned 2 kingdom classification, 3 kingdom, 4 kingdom, 5 kingdom and 6 kingdom classification. Okay. So who proposed these classification systems, and based on what they are going to classify these organisms? Let's see. So in brief, so 2 kingdom classification, it was proposed by the credit goes to either Aristotle or even Linnaeus system. Okay, they contributed for 2 kingdom classification. So according to them, the whole organisms are classified either into plants and animals based on the entire morphology, whether they look like plants or animals. Okay. So remember, at that time during this 2 kingdom classification, there was no concept of microorganisms. Okay. So ಅಲ್ಲಿ microscope invention ಆಗಿರಲಿಲ್ಲ, ಮತ್ತು microscopic organisms ಇರುತ್ತೆ ಅನ್ನೋ ಒಂದು ಕಾನ್ಸೆಪ್ಟ್ ಇರಲಿಲ್ಲ. So they classify the organisms into plants and animals based on their morphological characters. So later, with the advancement of science, there is a need to propose one more system of classification. That system of classification is 3 kingdom classification. Okay. So in a 3 kingdom classification, one more kingdom was introduced. Okay. So the new kingdom that was introduced in a 3 system classification is Protistans. Okay. So the new kingdom added is Protista, and it was proposed by Ernst Haeckel. Ernst Haeckel proposed a 3 kingdom classification, and he introduced a new kingdom along with plant and animals. Plant and animals. So during that time, microscope was invented and small organisms were also there. So those small organisms, they are distributed later, they are distributed into plant and animals also. Okay. So for those microorganisms, there is a need to create one more kingdom. The microorganisms which have the property of both plants and animals, they are grouped together into one category. That category is Protista. Okay. So Ernst Haeckel proposed Protista. Okay. So remember, Protista is exclusively for microorganisms. Okay. So it includes only microorganisms. Okay. Yeah. Next, with a little advancement of science again, there is a need to propose one more system of classification. Okay. So in that, with the advancement of science, the scientists know that microorganisms, so different kinds of microorganisms were there, and they have an incipient nucleus. Without a definite nucleus, there is organisms which have no nucleus. At the time, there was a concept was developed like prokaryotes and eukaryotes. Those who are organisms have true nucleus are eukaryotes, without a true nucleus they are prokaryotes. So in that sense, new kingdom was proposed. Okay. So new kingdom was proposed by Copeland. Copeland is a scientist who proposed a 4 kingdom classification, where he introduced one more kingdom. So Protista was retained as it is, and plante, actually it is a plante or plants and animals. Okay, so based on the character of the nucleus, new kingdom was created, that is Monera. Okay, so what are all the microorganisms which were earlier put under this Protista? ಯಾವುದು ಪ್ರೊಟಿಸ್ಟಾದೊಳಗೆ ಸೇರಿಸಿತ್ತೋ ಅದರಿಂದ ಕೆಲವೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ನ ತಗೊಂಡೋಗಿ ಇನ್ನೊಂದು ಕಿಂಗ್ಡಮಿಗೆ ಸೇರಿಸಿದ್ರು, ದಟ್ ಈಸ್ ಮೊನೆರಾ. ಓಕೆ, ಸಿ ದೇರ್ ಹಿಯರ್ ಮೊನೆರಾ ಈಸ್ ಎಕ್ಸ್ಕ್ಲೂಸಿವ್ಲಿ ಫಾರ್ prokaryotes. Exclusively, Monera is exclusive for prokaryotic unicellular organisms, whereas the Protista is exclusively for eukaryotic microorganisms. Okay, so 4 kingdom classification is proposed. With a little advancement in science again, there is a need to separate some organisms which are present in one group. They created a new kingdom. That kingdom was proposed by R.H. Whittaker. R.H. Whittaker's 5 kingdom classification. So Whittaker classified the plants. So here the most of the cases, in all the cases it based on microorganisms, classification of microorganisms are these plants and animals based on their morphology only. But here Whittaker, by considering one more thing, okay, so he considered whether they are heterotrophs or autotrophs. Okay, so based on that he classified or he introduced one more kingdom. So that kingdom is, so Monera is retained as it is, Protista is retained as it is, but a few organisms which are like new kingdom, but the organisms which are in Protista earlier, but they have the property of a new kingdom, they are shifted to this kingdom. So I will tell you that later. Then most earlier those organisms were present in plant kingdom. Okay, later Whittaker take those from plant kingdom and put them in a new kingdom. That new kingdom is Mycota. So remember earlier plants and fungi, they are in the same kingdom. ಯಾಕೆಂದರೆ ಈಗ ಮೈಕೋಟ ಫಂಗೈ ಏನಿದೆ, ಈ ಫಂಗೈನ ಮೊದಲು ನಾವು ಪ್ಲಾಂಟೇ ಕಿಂಗ್ಡಮ್ನಲ್ಲೇ ಓದ್ತಾ ಇದ್ವಿ. ಓಕೆ, ಸೊ ಲೇಟರ್ ವಿಟೇಕರ್ ಶಿಫ್ಟೆಡ್ ದೆಮ್ ಫ್ರಮ್ ಪ್ಲಾಂಟ್ಸ್ ಟು ನ್ಯೂ ಕಿಂಗ್ಡಮ್ ಕಾಲ್ಡ್ ಮೈಕೋಟ. ಓಕೆ, ಸೊ ದಿಸ್ ಇಸ್ ದ 5 ಕಿಂಗ್ಡಮ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್. ಸೊ ಇದಾದ ನಂತರ ಇನ್ನೊಂದು ಕಿಂಗ್ಡಮ್ ಕೂಡ ಪ್ರಪೋಸ್ ಆಯಿತು. ಸೊ ದಿಸ್ 6 ಕಿಂಗ್ಡಮ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ಅ 3 ಡೊಮೈನ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್. ಇಟ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ 3 domain classification. Okay, so under this 3 domain 6 kingdoms were placed. Okay, so let me brief about this 3 domain. So you know the taxonomical hierarchy starting from kingdom, division or phylum, class, order, family, genus, okay, and species. So this is taxonomical ranking or taxonomical hierarchy. So at present kingdom is the highest hierarchy, so it is the topmost category of classification. But the scientist who proposed this 6 kingdom classification, he created one more hierarchy, that is a domain. Okay, so under this domain he classified into kingdoms. Okay, that's why it is called as a 3 domain system of classification. Those 3 domains were, so it is strictly based on the molecular level structure, that is archaea, eubacteria and eukaryon. Okay, so these are the 3 domains he created: archaea, eubacteria and eukaryon. Okay, so this archaea and eubacteria, actually they split from Monera. Monera is split into 2, archaea and eubacteria, based on their specific nature. Archaeobacteria have a specific nature with respect to their cell wall structure also. Then eubacteria, so simply what he did, that 3 domain, let me write 6 kingdom classification. It was proposed by Carlos. Okay, okay, so let me write here. So it was proposed by Carlos. Carlos proposed a 6 kingdom classification. So what are those 6 kingdoms? So he proposed, he split Monera into Archaeobacteria. He split into Archaeobacteria, Eubacteria, okay, and remaining all Protista, mycota remained as it is. Protista, mycota, plantae and animalia. Okay, 1, 2, 3, 4, 5, 6. Okay, kingdom wise it is a 6 kingdom, domain wise 3 domains. So what he did is split the kingdom Monera into 2 again. That becomes 6 kingdom classification. So eukaryotes consisting of all the eukaryotes, Protista, mycota, plant and animalia, they are eukaryotes. Whereas this archaebacteria, eubacteria, they are prokaryotes, and they shifted them into 2 domains also. So archaebacteria, based on what he classified this archaea and eubacteria means, based on the sequencing of 16S rRNA. So this 16S rRNA is ribosomal RNA. So ribosome consisting of, in case of prokaryotes, it consisting of 5S rRNA, 16S rRNA and 23S rRNA. So these are the 3 ribosomal RNA. Among them, he considered 16S rRNA sequence. So 16S rRNA sequence is different from archaea and eubacteria. So based on that only created 2 kingdoms or 3 domains. Okay. So archaea consisting of all archaebacteria. All archaebacteria were grouped in archaea, and all true bacteria, they are grouped in eubacteria. Okay. Eukaryon, sorry, it is not eubacteria, eukaryon. Sorry, eukaryon was there, eubacteria. Okay, so true bacteria are grouped in eubacteria and archaebacteria are grouped under the domain archaea. Okay, so this is about the brief regarding the systems of classification. So 2 kingdom, plants and animals, 3 kingdom proposed by Ernst Haeckel. The new kingdom that was added by Ernst Haeckel is Protista. So Ernst Haeckel, he added Protista. So who introduced the kingdom Protista means it is Ernst Haeckel. Then Copeland. So Copeland introduced a new kingdom called Monera, exclusively for prokaryotes, whereas the Protista is exclusively for eukaryotic microorganisms. Okay, whereas Whittaker, R.H. Whittaker introduced the kingdom Mycota. Then Carlos created a new kingdom, that is Archaebacteria. So along with the Monera is considered as a eubacteria itself there. So like this, different systems of classification were proposed with the advancement in the science. Okay, so these organisms which comes under these different kingdoms have a peculiar character, and some have, there is no defined boundary for them. So especially with respect to Protista. So this Protista has the property of organisms which resembles the character like monerans also, sometimes they resembles the mycota also, and resembles plants also, so resembles animals also. So those who are present in Protista. Okay, so this is called like a mixed group. There is no definite boundary for, but Monera, mycota, plant, animalia, there is a boundary. If it look like this is the Monera, you can easily say. Mycota, if it has a chitinous cell wall, you can say it is fungi. Like that, there is a definite boundary. But for Protistans, you cannot give any definite boundary because it is a mixed group of organisms. Okay, so according to some evolutionary biologists, from Protistans only, all other groups like mycota, plant and animalia evolved during course of evolution. Okay, so this is a brief regarding the classification. So let's move on to the individual classification system. So we need to learn about a Whittaker system of classification. Okay, so 5 kingdom classification you need to learn in detail. Okay, so let's come to 5 kingdom classification. So 5 kingdom classification. So based on what Whittaker classify the organisms into 5 kingdom. Okay, so he consider cell or thallus nature. Okay, so he consider their reproduction. He consider cell wall material, and he consider whether they are autotrophs or heterotrophs based on okay, simply write mode of nutrition based on mode of nutrition. So these are the points under reproduction, mode of reproduction. So by considering these factors, he created the new kingdom, mycota. So mycota, they have mode of nutrition like heterotrophs. So why he separate them from plants means all plants are autotrophs, but fungi were heterotrophs. So this is the major reason he considered to shift fungi from the plants. Okay, yeah. Then reproduction. So fungi shows the reproduction types like asexual, sexual also. Okay, so asexual and sexual. In asexual, they reproduce by producing spores, sexually by oogametes, isogametes, and anisogametes, like the different methods they have. And a cell wall material, they have a specific cell wall material like chitinase. Okay, so reproduction already I told. Okay. So by considering these characters, Whittaker created a new kingdom called as fungi. Okay, so at the same time, in the blue-green algae, so blue-green algae, nostoc, anabina, they are all earlier in plant kingdom because they have chlorophyll. So all chlorophyll containing organisms were grouped in plant itself. But later, those nostoc, anabina, they are treated as a blue-green algae. They were shifted from a plant to monera, because those organisms, they lack definite nucleus. So definite nucleus irlilla anteli adana tagondogi innondu kingdom, that is monera ge seristara. Okay, so this is about the biological classification. Let's move on to the basic characters of moneras. Okay, so monera are the very specific group of organisms. First thing you need to be remembered, they are all prokaryotes. Monerans are prokaryotes. Okay, so they have the cell wall material. They have a cell wall material made up of peptidoglycan, or it is also called as murin. Okay, so this peptidoglycan or murin have a subunit called N-acetyl muramic acid and N-acetyl glucosamine. Okay, so amino groups kuda irutte cell wall alli. Murin they have made up of NAM and NAG. Okay, so ee riti peptidoglycan matte murin ittu andre they are treated as eubacteria. Actually eubacteria, and few organisms have different types of cell wall again called as a pseudopeptidoglycan, okay, or pseudomurin. The pseudomurin is present in archaebacteria. Okay, so based on the cell wall structure itself, we can classify them into archaebacteria and eubacteria. So archaebacteria are considered as a very early or primitive bacteria. So these archaebacteria have a special character. They can tolerate high range of temperature, high range of salinity. They can tolerate very low temperature as well. So those are the properties of archaebacteria. Then the cell membrane, the same kind. Cell membrane is made up of, so cell membrane. So here we are going to read eubacteria and archaebacteria under the same kingdom, because according to Whittaker's system of classification, both are comes under the same kingdom, Monera. So cell membrane, it is made up of a lipid and a protein as usual. All the living organisms are the same kind of composition. Then come to the cell organelles. So remember, cell organelles were absent. No cell organelle is present in the prokaryotes. So cell organelles are absent. So reproduction. Okay, so reproduction is by asexual method. Okay, so asexual method. In that, very common type of asexual reproduction in case of bacteria and archaebacteria as well is binary fission. Binary fission. So one individual cell undergo division called as a binary fission. So it is a most common type of asexual reproduction both in eubacteria and archaebacteria. Then sexual reproduction. So actual sexual reproduction is absent. So sexual method is absent, but Here there is no formation of gametes in sexual reproduction. No gamete formation occurs because ಯಾಕೆಂದ್ರೆ ಇಲ್ಲಿ ಡೆಫಿನೆಟ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲ. ಓಕೆ, ಸೊ ಅದಕ್ಕೆ ಇದನ್ನು ಪ್ರೊಕ್ಯಾರಿಯೋಟ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಪ್ರೊಕ್ಯಾರಿಯೋಟ್ಸ್ ಅಂದರೆ ನೋ ಡೆಫಿನೆಟ್ ನ್ಯೂಕ್ಲಿಯಸ್, ನೋ ಡೆಫಿನೆಟ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಆಬ್ಸೆಂಟ್ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇಸ್ ಸಿಂಪ್ಲಿ ಎಕ್ಸ್ಚೇಂಜ್ ಆಫ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್, ಸೊ ಕಾಲ್ಡ್ ಆಸ್ ಟ್ರಾನ್ಸ್ಡಕ್ಷನ್, transformation and conjugation. Okay, so transformation was experimentally proved by Griffith, then transduction by Hershey and Chase. You can remember this from second year part. Okay, so those are the reproduction type. And with respect to DNA, it is circular DNA. They have circular DNA. Okay, with no histone protein. No histone is present. No histone protein is present. Histone proteins ಅದರಲ್ಲಿ ಇರೋದಿಲ್ಲ. Okay, then okay, let me write here. No histone proteins are present. Then these eubacteria, they have with respect to their mode of nutrition, they are autotrophs and heterotrophs. ಸೊ ಎರಡು ಗ್ರೂಪ್ನಲ್ಲೂ ಇದೆ. ಸೊ ಆಟೋಟ್ರೋಪ್ಸು ಇರುತ್ತೆ ಮತ್ತು ಹೆಟೆರೋಟ್ರೋಪ್ಸು ಇರುತ್ತೆ. ಸೊ ಆಟೋಟ್ರೋಪ್ಸಲ್ಲಿ ಮತ್ತೆ 2 ಕ್ಯಾಟಗರೀಸ್, ಫೋಟೋಟ್ರೋಪ್ಸ್ ಆ್ಯಂಡ್ ಕೀಮೋಟ್ರೋಪ್ಸ್. ಓಕೆ, ಸೊ ಫೋಟೋಟ್ರೋಪಿಕ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ದೇ ಕ್ಯಾನ್ ಪರ್ಫಾರ್ಮ್ ಫೋಟೋಸಿಂಥಸಿಸ್. ಓಕೆ, ಫೋಟೋಸಿಂಥಸಿಸ್ ಮಾಡುತ್ತೆ. ದೇ ಯೂಸ್ ಕೆಮಿಕಲ್ ಆಸ್ ದ source. Okay, then the heterotrophs ಅಲ್ಲಿ there may be heterotrophs, they may be act as a parasites, they may be act as symbionts and saprophytes. Okay, so phototrophs, those who have chlorophyll like blue-green algae. So remember blue-green algae, earlier it was in plant, now they are in Monera because of they don't have definite nucleus. Okay, then chemotrophs, so those who use chemical as the source of energy. So chemotrophs example nitrifying bacteria, like a nitrifying bacteria. Okay, then a parasite which causes the disease, let me say salmonella, symbionts like rhizobium, then saprophytes which grows in the soil like a pseudomonas. Okay, so these are the group of bacteria based on their mode of nutrition. Okay, then so now bacteria are helpful in a different way. The milk curd, conversion of milk into curd, and we can obtain so many antibiotics from the bacteria itself. Like that, they have a very useful role also. I'm not going to discuss them now. Okay, yeah. So this is about the Monera's brief discussion. And come to the shape of a cell. So shape of a cell, bacteria have coccus, rod shaped called as bacillus, then comma shaped, okay, so vibrio, and spiral shaped, that is spirillum. Okay, so this coccus, it forms the chain called as a streptococcus. Similarly bacillus, a streptobacillus, so like that. And with respect to the flagella, so flagella they have, if they have single flagella, okay, monotrichous, If the two flagella on both the side, amphitrachus, tough flagella on one end, cephalotrichus. Okay, then flagella throughout the body, it is a peritrichus. So like this we can classify the bacteria based on the presence of flagella. So along with that, bacteria can be classified based on their cell wall material. They can be classified into two categories. Okay, so let me deal that one also. So bacteria can be classified into gram positive and gram negative depending on their amount of cell wall material and presence of membrane over the bacteria. So that can be classified into gram positive and gram negative. Gram positive and gram negative. Okay, if the bacteria has thick peptidoglycan, suppose this is a peptidoglycan, let me write down murin also, same name, murin or peptidoglycan is same, so it is a cell wall. This is one case. And in gram negative bacteria, this murin is very less. Murin amount is very less. So inside they have cell membrane. Okay, but gram negative bacteria, okay, so they have one more layer of membrane over there. Innondu layer membrane irutte, so it has membrane. So when you perform the gram staining bacteria, so during gram staining, gram positive bacteria, they took the first stain, they become purple. So purple color bantu andre gram positive bacteria, gram negative bacteria andre they appears pink or red. Okay, first stain ಯಾವುದು ಇರುತ್ತೆ ಅದೇ ಗಟ್ಟಿಯಾಗಿ ಕುತ್ಕೊಂಡು ಕಲರ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿಬಿಡುತ್ತೆ. So they become gram positive. So but ಈ ಕೇಸಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಸ್ಟೈನ್ ಕೂತಿರುತ್ತೆ, when you add alcohol, the color is removed, counter stain is going to be stain there. So they appears pink or red. So it depends on amount of murin. Gram positive have more murin. Remember one more name is peptidoglycan. So more peptidoglycan. Whereas gram negative, less murin. They have less murin. Okay, so this is how we can classify the bacteria into gram positive and gram negative. So amount of peptidoglycan is more means gram positive, amount of peptidoglycan is very less means gram negative. Okay, so this is about the kingdom Monera. Okay. So next kingdom, that is Protista. As I already told, Protista is the mixed group of organisms. Okay, so based on their characters, protistans can be classified. So most of them, they resembles the plant character also and animals character also. ಎರಡು ತರದ್ದು ಇದೆ. So this is a mixed group. There is no definite boundary for this protistans. Okay, so these protistans can be further classified into chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Okay, so protistans can be classified into this chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. So based on mode of nutrition, so chrysophytes are autotrophs. Okay, dinoflagellates, they are autotrophs. Okay, chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Okay, so euglenoids. Okay, so euglenoids, it is a mixotroph. It can act as both auto and hetero. Slime molds, they are pure heterotrophs. And protozoans also heterotrophs. Okay, here cell wall is made up of, in case of Garnulax, cell wall is made up of cellulose. Okay, then euglenoids. So example, euglena. Okay, so no cell wall, no cell wall. Instead, pellicle is present. Okay, so pellicle is a protein layer. It is protein layer. Okay, whereas the slime molds, so example, plasmophora, called as a plasmophora, no cell wall. Okay, but spores have cell wall. ಈ ಸ್ಪೋರ್ಸ್ ಅಲ್ಲಿ ಮಾತ್ರ ಸೆಲ್ ವಾಲ್ ಇರುತ್ತೆ. ಸೊ ಆರ್ಗ್ಯಾನಿಸಮ್ ನಲ್ಲಿ ಸೆಲ್ ವಾಲ್ ಇರಲ್ಲ. ಸೊ ಪ್ರೋಟೋಜೋವಾನ್ಸ್, ಎಕ್ಸಾಂಪಲ್ ಲೈಕ್ ಅಮೀಬಾ, ಸಮ್ ಸೆಲ್ ವಾಲ್ ಪ್ರೆಸೆಂಟ್, ಸೆಲ್ ವಾಲ್ ಇರುತ್ತೆ ಆ್ಯಂಡ್ ಆಬ್ಸೆಂಟ್ ಕೂಡ. ಓಕೆ, ಆಬ್ಸೆಂಟ್ ಕೂಡ ಇರುತ್ತೆ. ಸೊ ಪ್ರೋಟೋಜೋವಾನ್ಸ್, ಸೊ ಇಲ್ಲಿ ಅಮೀಬಾ, it has a silica, especially amoeba has silica depositions also. Okay, a cell membrane melgade silica irutte. Okay, so these protozoans can be further classified into different categories. So hope you understand the different types of cell wall materials. Okay, so chrysophytes have silica, dinoflagellates have cellulose, euglenids no cell wall, slime molds have no cell wall, but spore conditionally they have cell wall. So protozoans usually cell wall is absent, but a silica deposition is observed in amoeba. Okay, so this is about the cell wall material of these organisms. Okay, then another speciality is here. So chrysophytes, they have the cell wall like a soap box. They fix like a soap box tara fix agirutte. It is made up of silica. Okay, and they forms a diatomaceous earth. Diatomaceous earth. So this diatomaceous earth is used as a polishing agent, and even carborundum can be manufactured by using that diatomaceous earth. Then a dinoflagellate, Garnulax, which have a dinoflagellate. So there is a structure like this, cell plates. Between the two cell plates, one flagella is raised. So one vertical flagella, one horizontal flagella. So vertical flagella and horizontal flagella both are present in dinoflagellates. That's why the name dinoflagellate, eradu flagella anteli, which causes red tide. Okay, so innondu speciality ide. There is a species called Noctiluca, which is responsible for bioluminescence na cause ಮಾಡುತ್ತೆ. Noctiluca, which belongs to dinoflagellate. Okay. Okay, next come to euglenids. The only representative organism we can remember is euglena. So euglena, it has a slipper like appearance like this. It has a small flagella called as cilia. So idu cilia irutte idra melgade ella hinge. Sorry, I am talking about paramecium, not paramecium, euglena. So euglena has the structure like this. One short flagella, one long flagella it has, and it has a two nucleus they have. Okay, and vacuole, contractile vacuoles also they have. And the speciality of this euglena, it is considered as mixotroph. ಯಾಕೆ ಅಂದರೆ in the presence of light it acts as a plant, in the absence of light it acts as an animal. That's why it is called as mixotroph. Okay, ನೋಡಿ euglena ಎರಡು ಥರ ಪ್ರಾಪರ್ಟಿನೂ ಇರುತ್ತೆ. Next come to slime molds. Okay, slime mold example, plasmophora, which can cause the disease in of course it is a true heterotrophs. They are two heterotrophs. Okay, so this slime molds, that resembles fungi. It resembles fungi, but no cell wall. Okay, so ಸೊ ಅದರಲ್ಲಿ ಸೆಲ್ ವಾಲ್ ಇರೋದಿಲ್ಲ, ಬಟ್ ನೋಡೋಕ್ಕೆಲ್ಲ ಪ್ರಾಪರ್ಟೀಸ್ ಎಲ್ಲ ದೇ ರಿಸೆಂಬಲ್ಸ್ ಫಂಗೈ ಇಟ್ ಸೆಲ್ಫ್. ಓಕೆ, ಸೊ ಸೆಲ್ ವಾಲ್ ಪ್ರೆಸೆಂಟ್ ಓನ್ಲಿ ಇನ್ ದ ಸ್ಪೋರ್ಸ್. ಓಕೆ, ಅಗ್ರಿಗೇಷನ್ ಆಫ್ ಪ್ಲಾಸ್ಮೋಡಿಯಂ. ಓಕೆ, ಅದರದ್ದು ಇದನ್ನು ಪ್ಲಾಸ್ಮೋಡಿಯಂ ಅಂತ ಕರಿತೀವಿ. ಮೇನ್ ಪ್ಲಾಂಟ್ ಥಾಲಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ ಪ್ಲಾಸ್ಮೋಡಿಯಂ. ಸೊ ಪ್ಲಾಸ್ಮೋಡಿಯಂ ಅಗ್ರಿಗೇಟ್ ಆಗಿ ಫಾರ್ಮ್ fruiting body. Fruiting body. That fruiting body produces spores. That spores contains cell wall. Okay, so it is a true heterotrophs. Wooden logs melella ಜಾಸ್ತಿ ಬೆಳ್ಕೊಳ್ಳುತ್ತೆ, which are different colors they have. Then come to protozoans. Okay, so protozoans can be further classified into amoeboid, then ciliate, then flagellate. Okay, then sporozoans. Okay, so flagellate, sporozoans. Okay, yeah, so like that we can classify them: amoeboid, ciliate, flagellate and sporozoans. So amoeboid example, entamoeba or amoeba. Okay, so ciliated ನೋಡಿ ಈ ಥರ ಸ್ಲಿಪ್ಪರ್ ಲೈಕ್ ಇರುತ್ತೆ, ಇದೆಲ್ಲ ಸೀಲಿಯಾ ಇರುತ್ತೆ ಸುತ್ತ. ಸೊ ಪ್ಯಾರಾಮೀಷಿಯಂ is example for that. Then flagellate example, trypanosoma. Trypanosoma causes sleeping sickness disease and was vector is a sese fly. Then sporozoans, protozoans. So amoeboid, protozoans, then sporozoans. Malarial parasite, plasmodium. Okay, so infectious spore-like stage is present in the sporozoans called as plasmodium. So these are the characters of protistans. Okay, so protistans, there is no definite characters can be fixed for this because they have a mixed group of organisms. Okay, so this is about protistans. So next come to one more group of organisms, that kingdom. So next kingdom is after Monera protista is fungi. So fungi is the kingdom created by Whitaker based on their pure nature with respect to their reproduction mode of nutrition. So fungi are cosmopolitan, they found everywhere. So based on the nature, they have a thin slender thread-like structure that is called a hyphae. Okay, so this hyphae is of two types. Tube-like structure irutte. Some have the structure like this, compartment they have, and some have no compartment with a multinucleate condition. So if cross wall is present, it is called a septate. Without cross wall, it is aseptate. Okay, so here aseptate multinucleate condition is there called as a cenocytic. Okay, septate may have single nuclei or multinuclei, and they are haploid in nature. Remember this, fungi are haploid in nature. So based on hyphal nature and how they reproduce, fungi can be classified into 4 classes. Phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. Okay, so this is how we can classify the fungi based on their mode of reproduction, purely based on mode of reproduction. So phycomycetes have aseptate hyphae. They have aseptate hyphae. Ascomycetes have septate hyphae. Basidiomycetes also have septate hyphae. So deuteromycetes also have septate hyphae. So here cenocytic hyphae or aseptate hyphae is a primitive character. So phycomycetes are primitive fungi. Okay. So let me deal with the reproduction. So what type of reproduction they undergo? Let's see in brief. Okay. So mode of reproduction. So phycomycetes can reproduce by asexual method as well as sexual method. In both the type they undergo reproduction. So asexual method is isogamous, sorry, not isogamous, here sexual is iso or mostly they are oogamous. Let me write directly, they are oogamous type. So oogamous means a non-motile larger female gamete and motile small male gamete. So asexually by producing spores, haploid spores, they are going to be reproduced. Okay. And ascomycetes. Okay. So ascomycete asexually they reproduce by conidia or endogenous spores. Okay, so conidia or spores, let me write in general only. So the spores, in case of fungi there are two kinds of spores. Okay, so sometimes spores are produced within a sac. ಈ ರೀತಿ spores produce ಆಗ್ತಾ ಇದ್ರೆ, so these are called sporangiospores. Sporangiospores. If the spores are born externally directly, so ಈ ರೀತಿ spores direct external born ಆಗಿದ್ರೆ, ಈ ರೀತಿ born ಆಗಿದ್ರೆ, they are called conidia. Okay, so conidia ಮತ್ತೆ sporangiospore ಎರಡು ಇರುತ್ತೆ. Okay, so spores in the sense usually they are sporangiospores itself. Okay, so spores ಆಗುತ್ತೆ. Whereas in sexual reproduction they produce ascospores. They produce ascospores. So ascospores, ascomycete. So basidiomycetes. So in basidiomycete asexual reproduction is not seen, whereas of course the only method we can observe is fragmentation. There is no specific spore-like structures are formed in basidiomycetes, whereas in sexual reproduction they are reproduced by basidiospores. They reproduce by producing basidiospores, whereas in deuteromycetes all are reproduced by producing conidia. So sexual reproduction is absent. So sexual reproduction is absent in deuteromycetes. That is the reason why they are called as imperfect fungi. They are called as imperfect fungi. Okay, so let me deal with some specific characters with respect to ascospores. Ascospores are produced within ascus. So the fruiting body anta kariತೀವಿ ಅದಕ್ಕೆ, fruiting body in the structure, they are produced within ascus. So ascus is like this and it produces exactly 8 spores. 1 2 3 4 5 6 7 8 spores is going to produce, whereas basidiospore are produced in basidia, and each basidia can produce exactly 4 spores. 1 2 3 4. So these are basidiospores and these are ascospores. Okay, so compactly arranged ascospore, so ascus is called as ascocarp. Fruiting body is called ascocarp. Similarly basidiomycete fruiting body is called basidiocarp. Okay, so in deuteromycetes sexual reproduction is absent. So best example for basidiocarp is mushroom. So mushroom is the best example for basidiocarp. Is a all the mycelar group put together to form a structure called basidiocarp. Okay, so this is about the simple thing regarding all the fungi classes. And one more important thing you need to remember in the sexual reproduction, especially ascomycetes and basidiomycetes, there is a stage called dikaryon. Okay, so dikaryon is a state where in fungi during sexual reproduction. Okay, so let me take the example like this. So 3 different types of hyphae I am writing. If both antheridia and archegonia are born on the same hyphae, ಎರಡು ಕೂಡ ಒಂದೇ ಕಡೆ born ಆಗಿದ್ರೆ, suppose this is female reproductive archegonia, if it is antheridia, it is called homothallic. ಇದನ್ನು ಹೋಮೋಥ್ಯಾಲಿಕ್ ಅಂತೀವಿ. ಸೊ ಒಂದು ಫಂಗೈನಲ್ಲಿ ಒಂದು ಐಫೆನಲ್ಲಿ it produces antheridia, in another it produces archegonia like this. So this is called heterothallic. ಓಕೆ, ಸೊ ಈ ರೀತಿ ಎರಡು ಕಂಡೀಷನ್ ಇದೆ, ಹೋಮೋಥ್ಯಾಲಿಕ್ ಮತ್ತು ಹೆಟಿರೋಥ್ಯಾಲಿಕ್. ಓಕೆ, ಸೊ ಫೈಕೋಮೈಸಿಟ್ಸ್, ನಾನು ಆಗಲೇ ಹೇಳಿದೆ ಹೂ ಗ್ಯಾಮಸ್, ಐ ಮಿಸ್ಡ್ ಒನ್ ಥಿಂಗ್ ದೇರ್. ಸೊ ಇನ್ ಫೈಕೋಮೈಸಿಟ್ ದೇ ಪ್ರೊಡ್ಯೂಸ್ zygospores. They produce zygospore as a sexual spores. So Phycomycete zygospore, Ascomycetes ascospore, Basidiomycetes basidiospores, whereas Deuteromycete no sexual spores. Okay, so during sexual reproduction between two different hyphae, let me take. So one produces archegonia like structure, septate hyphae. At the point of contact, a wall is dissolved. So here individually have haploid spores at the wall of contact the wall contact agire kade, whole the thing of this compartment move this side along with the cytoplasm. ನೋಡ್ಕೊಳ್ಳಿ ಇಲ್ಲಿ ಕೇರ್ಫುಲ್ ಆಗಿ, ಇಲ್ಲಿರೋ ಸೈಟೋಪ್ಲಾಸಮ್ ಸಮೇತ ಆ ಸೆಲ್ ಒಳಗೆ ಹೋಯ್ತು. ಓಕೆ, ನಾವು ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ ಡಿಲೇಸ್, ಬಟ್ ಪ್ಲಾಸ್ಮೋಗ್ಯಾಮಿ occurs immediately. Plasmogamy occurs immediately. At the point of contact cell wall dissolve ಆಗುತ್ತೆ, cytoplasm ಸಮೇತ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗೆ ಹೋಗುತ್ತೆ. ಸೊ ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ ಅಂದರೆ ಪ್ಲಾಸ್ಮೋಗ್ಯಾಮಿ occurs immediately, but ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ ಡಿಲೇಸ್. ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ ಇಮಿಡಿಯೇಟ್ ಆಗಿ ಆಗಲ್ಲ. ಸೊ ಇಟ್ ಕಂಟೈನ್ಸ್ ಟೂ ಡಿಫರೆಂಟ್ ನ್ಯೂಕ್ಲಿಯೈ. ದ ಸ್ಟೇಜ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಡೈಕ್ಯಾರಿಯಾನ್. The stage is called as dikaryon stage. So after some time fusion takes place. Again meiosis occurs, then forms 4 spores. They undergo mitosis again to form 8 spores. Now this structure developed into ascus. Okay, so each developed into spore like structure like this. Spores are going to be produced. So this is about the reproduction type. It is, remember, dikaryon is a common in Ascomycete as well as Basidiomycetes. Okay, so let me give some examples. Okay, so examples, and let me take 2 categories. One is for saprophytes, another parasite, ಅಂದರೆ ವಿಚ್ ಕ್ಯಾನ್ ಕಾಸ್ ದ ಡಿಸೀಸ್. ಎರಡನ್ನ ಎಕ್ಸಾಂಪಲ್ ಆಗಿ ತಗೊಳ್ಳೋಣ. ಸೊ ಫೈಕೋಮೈಸಿಟ್ಸ್ಗೆ ಸ್ಯಾಪ್ರೋಫೈಟಿಕ್ ಎಕ್ಸಾಂಪಲ್ ರೈಸೋಪಸ್, ಕಾಮನ್ಲಿ ಕಾಲ್ಡ್ ಆಸ್ ಬ್ರೆಡ್ ಮೋಲ್ಡ್. ಓಕೆ, ಸೊ ಪ್ಯಾರಾಸೈಟ್ ಆಲ್ಬಿಗೋ ಕಾಸಸ್ ವೈಟ್ ರಸ್ಟ್. Albigo causes white rust is the best example for parasitic Phycomycete. Ascomycete saprophytic is Penicillium, then Aspergillus. So these are example for Ascomycetes fungi which are saprophytic in nature, and even you can take the example of yeast. Okay, so parasitic Claviceps purpurea is a parasite which cause the disease in plants. Then Basidiomycete saprophytic is mushroom. So common mushroom, that is Agaricus. It is Agaricus species, and a parasite, that is Puccinia. Puccinia causes rust. So Claviceps causes the smut disease, and Puccinia causes rust disease. Okay, so it causes rust disease. Okay, so Deuteromycete saprophyte trichoderma, and for parasite Coletotrichum that causes red rot of sugarcane. Then let me take one more example, that is Alternaria that causes early blight. So these are the certain example for fungi. Okay, so that's it. So next move on to the kingdom plantae. So kingdom plantae, they have true cellulosic. There is a separate chapter on them, so you can learn detailed character in the chapter. Then next kingdom is Animalia. There is a special new chapter is on that kingdom, so you can learn the character there. I am not going to deal on that now. So next come to special group of things which are not included in any kingdom. So those are virus. So virus are nucleoproteins. Nucleoproteins, they are made up of nucleic acid as well as protein. Okay, the first scientist who isolated this virus were Davanagere. So he isolated and named tobacco mosaic virus. Okay, so this is the first virus which was detected. So tobacco mosaic virus is a rod shaped virus. Rod shape. Okay, so it has a protein coat. So along with this protein coat, so in the center it has the genetic material. So your genetic material is single stranded RNA. This protein coat is called capsid. So protein part of the virus is called as capsid. Individual one is called as capsomere. Capsomere. Okay, so this TME was crystallized by. Virus can be crystallized. So who showed that property, Indra? It is the Stanley. So Stanley crystallized the virus for the first time. Okay, so virus have the property. Inside the organism it acts as a living entity. Outside the organism it acts like a non-living entity. So crystallized by Stanley. Okay. Okay. Then so viruses are called as contagium, vivum, fluidum by Beijerinkia called them as contagium, vivum, fluidum. Andre poisonous fluid anteli. A poisonous fluid na tagondu yarige hakidru matte disease barutte anteli. So contagium, vivum, fluidum. So virus as a nucleoprotein. So you study the structure of bacteriophage. It has a head, then collar, contractile sheath it has, then a tile plate, tile fibers, central core has a pointed structure, infection peg, and it has a DNA as a genetic material. Okay, it's called bacteriophage, which infect the bacteria. Okay, so this is about the virus. So next we study the lichens. So lichens have mutually associated between fungi plus algae. Okay, so fungi ಮತ್ತು algae ಎರಡು ಒಟ್ಟಿಗೆ associate ಆಗಿರೋ organism is lichen. So fungi give the nutrients, in turn algae give the food. So fungal partner is mycobiont and algal partner is phycobiont, and this lichens are pollution indicators. Okay, so pollution indicate ಮಾಡುವಂಥ organisms ಇದು. Okay, so this is about lichens. So lichens can be of three types based on their thallus nature. One is crustose, foliose, and fruticose. So next is viroids. So viroids are the virus which lacks genetic material. Okay, sorry, which lacks protein code. Okay, similarly prions were also there. Okay, so prions infectious, here in viroids infectious genetic material. Genetic material is infectious here, whereas in prions infectious is protein coat. Okay, protein coat in a disease cause ಮಾಡುತ್ತೆ in case of prions. Viroids ಎಲ್ಲ ಆದರೆ genetic material. So viroids ಅಲ್ಲಿ no protein coat, prions ಅಲ್ಲಿ no genetic material. Okay, so viroids can cause us potato spindle tuber disease. Okay, so these are identified by Diner. Okay, so Diner identified potato spindle tuber disease causes viroids. So prions can cause mad cow disease. Okay, so this is about the viroids and the prions. So based on this, okay, start the competitive part. Let's see. Okay, so far we learned briefly regarding the chapter biological classification. Let's move on to the questions now. So select, actually it should be incorrect. Select incorrect match with a reference to Whittaker's system of classification. So option 1, Monera, unicellular and multicellular, autotrophic, decomposing, true cellulosic cell wall. Protista, unicellular, eukaryotic, photoautotrophs. Fungi, multicellular, loose tissue, eukaryotic, chitinous wall. Archaea, smallest, pleomorphic, pseudomulinic. Okay, so if you consider all the characters, so Protista character is right, they are unicellular, right, they are eukaryotic, they are photoautotrophs, and even decomposers are also there. In fungi, it is maybe unicellular and multicellular, loose tissue called as pseudoparenchyma, then they are eukaryotic and they contains chitin wall. Where Archaea, smallest organisms, then they are pleomorphic also, then they are pseudomulinic. So murin is a pseudomulin, so called pseudomulinic. Okay, so then Monera, they are unicellular of course, but no multicellular. Autotrophs okay, decomposing okay, true cellulosic wall is not there. So the incorrect option for this is option 1. Okay, so hope you got it. Yeah, let's move to the next question. So, which of the hyperthermophilic organisms that grow in high temperature habitat belong to this group? So, hyperthermophilic, and they can tolerate very high temperature. Okay, so only the group of bacteria called as archaebacteria or archaea have the ability to tolerate high temperature as well as very low temperature as well, and they are methanogens also because they have the wall made up of pseudomurin. Due to the presence of pseudomurin, they can tolerate high temperature and even low temperature. Okay, so eubacteria, archaebacteria. So you learnt in the second year itself one of the bacteria which belongs to this archaebacteria, which has the thermostable polymerase enzyme is isolated from that. So example for archaea is Thermus aquaticus. The enzyme is a Thermus aquaticus isolate ಮಾಡ್ಕೊಂಡಿರುವಂತದ್ದು TAC polymerase. Okay, so you learned that. Then mycoplasma, they are the smallest. So I missed there in the synopsis. So mycoplasma, they are the smallest known organisms without a cell wall, and they are pleomorphic. So pleomorphic are the organism character where they can change the shape. Sometime they were like this and they become like this, or they become like this. So whatever the shape they want, they can take. Okay, ಯಾವ ಶೇಪ್ ಬೇಜಾರಾಗುತ್ತೋ ಆಮೇಲೆ ಇನ್ನೊಂದು ಶೇಪಿಗೆ ಶಿಫ್ಟ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ. So that is called pleomorphic. The pleomorphic nature of this mycoplasma is due to absence of cell wall. So cell wall illa, that is advantage for this mycoplasma. So they can change the shape. So pleomorphic andre can change the shape. Can change the shape. Anta group of organism pleomorphic anta ಹೇಳ್ತೀವಿ. And it is strictly anaerobic. Mycoplasma is anaerobic organism. Okay, smallest known cell wall less organism. Okay, is mycoplasma. Okay. Yeah, so next question. So in eubacteria, a cellular component that resembles eukaryotic cell, ಅಂದರೆ ಬ್ಯಾಕ್ಟೀರಿಯಾದ ಯಾವ ಕಂಪೋನೆಂಟು ಯೂಕ್ಯಾರಿಯೋಟಿಕ್ಗೆ ಮ್ಯಾಚ್ ಆಗುತ್ತೆ. Okay, so plasma membrane, nucleus, plasmid, cell wall. Okay, so nucleus ಒಂದರಲ್ಲಿದೆ ಒಂದರಲ್ಲಿಲ್ಲ. Eubacteria has a nucleus, eukarya has a nucleus, and bacteria no nucleus. Plasmid only bacteria have. Then cell wall, different materials were there. If you consider fungi, chitin. If you consider fungi, plant, it is cellulose, and in case of bacteria it is murin. So there is no match with this. Okay, so only match is plasma membrane. The plasma membrane which is present in the organisms, all the living organism is same. Okay, lipid and protein in the agiratha. Okay, let's move to the next question. Yeah, so genophores, the DNA in prokaryote, DNA and histone in prokaryote, DNA and protein, RNA in prokaryote. So genophore. So in case of bacteria, wherever that genetic material is present, that genetic material is called as genophore. ಅದನ್ನೇ genophore ಅಂತ ಕೂಡ ಹೇಳ್ತೀವಿ, ಅದನ್ನ ನ್ಯೂಕ್ಲಿಯಾಯ್ಡ್ ಅಂತ ಕೂಡ ಹೇಳ್ತೀವಿ. ಓಕೆ, ಸೊ genophore is DNA in prokaryote. ಸೊ ಎಲ್ಲೆಲ್ಲಿರುತ್ತೋ ಆ ಜಾಗನ genophore ಅಂತೀವಿ, ಅದನ್ನ ನ್ಯೂಕ್ಲಿಯಾಯ್ಡ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ರೈಟ್ ಆಪ್ಷನ್ is DNA in prokaryote is called as genophore. And remember, histone proteins are absent in prokaryotes. Histones iralla adralle. So next question. So dinoflagellates, flagella lies longitudinally and other transversely in a furrow between. So in case of dinoflagellate, one flagella is present, that is vertical, and one more flagella which grows horizontally like this, then will become vertical. Okay, ಎರಡು ಫರ್ರೋಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಆ ಫರ್ರೋಗಳ ಮಧ್ಯೆ ಸಲ್ಕಸ್ ಅಂತ ಕೂಡ ಹೇಳ್ತೀವಿ, ಆ ಸಲ್ಕಸ್ ಮಧ್ಯೆಯಿಂದ ಈ ಥರ ಫ್ಲಾಜೆಲ್ಲ ಬಂದಿರುತ್ತೆ. ಓಕೆ, ಸೊ ಎರಡು ಸೆಲ್ಯುಲೋಸಿಕ್ ವಾಲ್ ಪ್ಲೇಟ್ಸ್ ಮಧ್ಯೆ, ದಿಸ್ ಇಸ್ ಒನ್ ಸೆಲ್ಯುಲೋಸಿಕ್ ವಾಲ್ ಪ್ಲೇಟ್, ದಿಸ್ ಇಸ್ ಅನದರ್ ಸೆಲ್ಯುಲೋಸಿಕ್ ವಾಲ್ ಪ್ಲೇಟ್, ಇವೆರಡರ ಮಧ್ಯೆ ಫ್ಲಾಜೆಲ್ಲ ಬಂದಿರುತ್ತೆ. ಸೊ ಇಟ್ ಹಸ್ ಟೂ ಟೈಪ್ಸ್ ಆಫ್ ಫ್ಲಾಜೆಲ್ಲ. ಸೊ ಇಟ್ ಈಸ್ ಸೆಲ್ಯುಲೋಸಿಕ್ ವಾಲ್ ಪ್ಲೇಟ್ಸ್. ಓಕೆ, next question number 6. So kingdom with a polysaccharide plus amino acid and only polysaccharide cell wall R. So kingdom with the polysaccharide plus amino acid ಜೊತೆಗೆ ಬರೀ polysaccharide. So polysaccharide ಶುಗರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಒಂದು ಜಾಯಿನ್ ಆಗಿ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಆಗಿರುತ್ತೆ. ಸೊ ಪಾಲಿಸ್ಯಾಕರೈಡು ಇರುತ್ತೆ ಪ್ಲಸ್ ಅಮೈನೋ ಆ್ಯಸಿಡು ಇರುತ್ತೆ. ಸೊ ಆ ರೀತಿ ಇರುವಂಥದ್ದು ಗ್ರೂಪು. ಓಕೆ, ಸೊ ಈ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕ್ಯಾನ್ನಲ್ಲಿ ಐ ಆಲ್ರೆಡಿ ಟೋಲ್ಡ್ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕ್ಯಾನ್ ಹ್ಯಾಸ್ ನ್ಯಾಮ್ ಆ್ಯಂಡ್ ನ್ಯಾಗ್ ಅಂತ ಹೇಳಿದ್ದೆ. ಎನ್ ಅಸಿಟೈಲ್ ಮ್ಯೂರಾಮಿಕ್ ಆ್ಯಸಿಡ್, ಎನ್ ಅಸಿಟೈಲ್ ಗ್ಲುಕೋಸ್ ಅಮೈನೋ ಗ್ರೂಪ್ ಇರುತ್ತೆ. ಸೊ ಅಮೈನೋ ಗ್ರೂಪ್ ಲೆಕ್ಕಕ್ಕೆ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಅಂತೇಳಿ. ಸೊ ಆ ರೀತಿ ಅಮೈನೋ ಆ್ಯಸಿಡ್ ಇರುವಂಥದ್ದು ಯಾವುದು ಅಂದರೆ ಮೊನೆರಾನ್ಸಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಜೊತೆಗೆ ಓನ್ಲಿ ಪಾಲಿಸ್ಯಾಕರೈಡ್. ಸೊ ಓನ್ಲಿ ಪಾಲಿಸ್ಯಾಕರೈಡ್ ಅಂದರೆ ಸೆಲ್ಯುಲೋಸ್. ಸೊ ಸೆಲ್ಯುಲೋಸ್ ಇಸ್ ಎ ಪಾಲಿಮರ್ ಆಫ್ ಶುಗರ್. ಇಟ್ ಇಸ್ ಎ ಪಾಲಿಮರ್ ಆಫ್ sugar. So plantae. So Monera and plantae are the examples for polysaccharide amino acid containing Monera. Only polysaccharide is plantae. Okay. So next. Then who proposed the four kingdom classification? So four kingdom classifications were proposed by Copeland, who introduced Monera, Ernst Haeckel 3 kingdom classification, Linnaeus 2 kingdom classification. Theophrastus is called as father of botany. Okay, so Copeland introduced Monera, so 4 kingdom. Haeckel introduced Protista. Okay, so Linnaeus 2 kingdom classification. Because Linnaeus strictly followed the Aristotle kingdom classification. Okay. Next question. Yeah. So respiratory enzymes are found associated with the plasma membrane. Okay. So in case of bacteria, there is no specific organ for respiration like in mitochondria. So instead, that bacterial membrane shows the foldings. Okay. On this folded membrane itself, enzymes which are responsible for respiration are present. Okay, so those are called a mesosome. A mesosome in the folded membrane, it's itself a sites for respiration. Okay, yeah, so it is a Clostridium is a bacteria. So Clostridium is the answer for this. Chlorella is for algae, Cara is algae, Chlamydomonas is algae. So algae nalli specific mitochondria irutte for respiration. So Clostridium is the answer for this. Next. Okay. Wait, so next. Yeah. So group of bacteria, shape based on the shape. Coccus is spherical, Bacillus is rod shaped, Spirillum is spiral, Vibria is comma shaped. Okay, so A2, B1, then spirillum C3, so no, so idu modle eliminate agittu, so right one is second. So Vibram 5. So option for that right answer, sorry, this is 4. Yeah, so Vibrium comma shaped. So right answer is second one. Right, what is the next question? Question number 10. So how many of the given features are associated with the incipient nucleus of Monera, like a naked DNA? So naked DNA, incipient nucleus, it's right. RNA, they have RNA. Non-histone protein is, they have non-histone protein. Histone proteins are absent. Then a linear single-stranded DNA is absent since it is circular DNA. So circular double-stranded DNA is present. So totally 4 were there. So this is the right answer. Okay. So hope you got it. So read the question, then answer it. So you can pause and read the question carefully, then you can answer. Okay. Next question. So heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom. So heterotrophs ಆಗಿರಬೇಕು, they are unable to prepare their own food. Eukaryotic ಆಗಿರಬೇಕು, multicellular organisms ಜೊತೆಗೆ cell wall ಇಲ್ಲದೆ ಇರೋ organism ಅಂದರೆ. So among these, protista have cell wall, planta have cell wall, fungi have cell wall. So right option is this, animalia. So protista has different types of cell wall materials like cellulose ಇರುತ್ತೆ, then silica ಇರುತ್ತೆ, whereas fungi chitinous, plant has cellulose. So animalia is the answer for this question. Okay, let's go to the next question. Yeah. Select the incorrect statement. Nostoc and Anabina have heterocyst for nitrogen fixation. So both Nostoc and Anabina, so they have a specialized cells along with the vegetative cell. They have specialized cell like this called as heterocyst. So this heterocyst are responsible for conversion of nitrogen into ammonia. So nitrogen fixation ಎಲ್ಲಾಗುತ್ತೆ ಅಂದರೆ heterocystal ಆಗುತ್ತೆ. And ಇಲ್ಲಿ this is a site where remaining all cells, they have normal vegetative cells, they can perform photosynthesis. Yes, right? Then cyanobacteria often form blooms in polluted water bodies. So water bodies ಮೇಲೆ green color mat ತೇಲ್ತಾ ಇರುತ್ತೆ, green color mat ಥರ. So it is cyanobacteria. When a nitrogen content is increased in the soil, water body phosphorus ಮತ್ತು nitrogen content increase ಆದಾಗ this bloom ಆಗುತ್ತೆ called as algal bloom. Okay, it is also due to cyanobacteria. So heterotrophic bacteria are more abundant in nature. Obviously heterotrophic bacteria ಜಾಸ್ತಿ ಇದೆ instead of autotrophic bacteria. So autotrophic bacteria are very less in number and heterotrophic bacteria are abundant. Okay, the cell wall of mycoplasma are made up of chitin. So mycoplasma, they don't have cell wall at all. So this is the wrong one. So incorrect option is option 4. Okay, let's come to the question number next. Okay. So mark the odd one with respect to cell wall. So halophiles have pseudomurine, methanogens have pseudomurine, thermoacidophiles have pseudomurine. So no, pseudomurine, it is a pseudomurine. Cyanobacteria, so cyanobacteria have a specific type of murine, that is only murine. They have murine cell wall, and these three belongs to archaebacteria, and this belong to blue green algae, cyanobacteria. So it's not the answer. Come to okay. Next, which of the following is correct? All slime molds are haploid. Not all slime molds are haploid. Protozoans lack cell wall. Dinoflagellates are non-motile. Pellicle is absent in euglena. Okay, so which one is correct? So all slime molds are haploid, diploid irutte. So it is not the one. Protozoans lack cell wall. Yeah, protozoans like amoeba lacks a cell wall. That is right. Dinoflagellates are non-motile. They have motility. They shows motility. Pellicle is absent in euglena. Euglena present. Pellicle is present in euglena only, the protein layer. Okay. So second is the right option. Next question. Yeah. So Gonulax, Noctiluca, Neurospora, Agaricus, bioluminescence, biochemical and fruiting body, rapid multiplication. Okay. Okay. So Gonulax, it shows the rapid multiplication. Okay. So it shows the rapid multiplication. I think option is missing. So option is missing here. Sorry. So Gonulax, rapid multiplication. Noctiluca shows the bioluminescence, whereas Neurospora, acetaldehyde, some chemicals are produced from Neurospora. Then Agaricus is a fruiting body. Okay, so this is the match the following. Sorry, options are missing by mistake. So preparing slide, something is missed. Okay. So next, question number 16. Protists reproduce asexually and asexually by involving these process respectively. In case of a protista, so how the sexual reproduction is? Cell fusion and zygote formation. So if a unicellular are there, if haploid cells are there, their cell itself they are fused together called a cell fusion, and a zygote is going to be formed. Cell fusion agata, zygote is going to be formed. Fission agala. Zygote formation, cell fission. No, main you have to consider zygote formation and cell fusion. Cell fusion agate haploid content, then it forms the zygote. So this is a mode of reproduction in case of protista. Okay. Go to the next question. Okay. So let's see the statements. Yeast is a multicellular fungus. Okay. So yeast is not a multicellular, it is the unicellular fungi. It is unicellular fungi which do not produce hyphae. This is the only fungi without mycelia. Okay. Then penicillium is a unicellular fungi. So penicillium is not a unicellular. So penicillium is a multicellular. It has that multicellular compartments like this. Either compartments irutte. Then it can reproduce by producing the conidia. So it is not a multicellular. So albugo is a parasitic fungus on mustard. Yes, it is a parasitic on mustard which causes white rust. Okay. So only statement 1 and 2 statement are correct? No. All the above statements are incorrect? No. Only statement 3 is correct? Yes, this is the right option. Both the statement 1 and 3 are correct? No. So statement 3 is correct. Okay. Next come to the next question. So question number 18. So the sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different genotypes. It is the future of the sex organs are absent. Okay. See, sex organs, the specific sex organs antare ಆ organism ನಲ್ಲಿ ಏನು ಇರಲ್ಲ, but plasmogamy is brought about by the fusion of two vegetative or somatic cells. Okay. So ಎರಡು ಹೈಫೆಗಳು ಕೂಡ ಫ್ಯೂಷನ್ ಆಗೋ ಚಾನ್ಸ್ ಇರುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ಒನ್ ಹೈಫೆ, then this is another hyphae, whether come in contact, dissolve agata ball, then karyogamy occurs, that delays the plasmogamy occurs, karyogamy delays. Okay. Two vegetative or somatic cells of different genotypes, it is future of ಈ ರೀತಿ ಫ್ಯೂಷನ್ ಆಗಿ it developed into spores. 4 spores are going to be formed. So this is the future of. So there is no, ಇದು important in particular, specific sex organs are absent. Okay, in case of basidiomycetes. Okay, but the fructification takes place to produce a fruiting body. Okay, so that fruiting body produces basidiomycetes. Okay, so remember, basidiomycetes are the best examples for this answer. Next question number 19. So different organisms with their different locomotory structures were given. Let's see. So locomotory structures and protozoans. So pseudopodia, false fruit. Okay, ಯಾವ್ದ್ರಲ್ಲಿ ಇರುತ್ತೆ ಅಂತ ಗೊತ್ತಿರುತ್ತೆ ನಿಮಗೆ. So false fruit is found in Entamoeba histolytica. Okay, so A5 search for it is 4 A4. So A4 2 options were there. Then you can eliminate them. Okay. So cilia, flagella lack a locomotory structures. The one which lacks a locomotory structure, it is Plasmodium D3. Okay, so D3 there. Then go for cilia. So cilia present in Paramecium. Okay, so B1. So you can choose B1. So this is the right answer then. Okay. Then come to C2. Flagella, it is a trypanosoma. So trypanosoma contains flagella. So this trypanosoma causes sleeping sickness, and it was spread by sese fly, the fly called sese fly. Okay, so ಇದಕ್ಕೆ ವೆಕ್ಟರ್ ಇದು sese fly is a vector for trypanosoma. Trypanosoma ಗೆ ವೆಕ್ಟರ್ ಇದು sese fly. Okay, so this is about the locomotory structures and some protozoans. Okay, next come to the next question, the question number 20. Select the wrong statement. The term contagium vivum fluidum was coined by Bergerac. So that is right. So it is right. Then mosaic disease in tobacco and AIDS in human are caused by virus. Okay, so mosaic disease in tobacco, that is tobacco mosaic virus, and AIDS HIV. So both are virus. That's right. The virides were discovered by DJ Ivanovsky. No, virus were discovered by Ivanovsky. Virides were discovered by Diner. Okay. So W.M. Stanley showed that the viruses could be crystallized. Yeah, that's right. So ಎರಡು ಕರೆಕ್ಟ್ ಆಗಿದೆ, ರಾಂಗ್ ಆಗಿರೋದು ರಾಂಗ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಯಾವುದು ಅಂದರೆ ದಿಸ್ ಥರ್ಡ್ ಒನ್. ಸೊ ಥರ್ಡ್ ಒನ್ ಇಸ್ ದ ರಾಂಗ್ ಸ್ಟೇಟ್ಮೆಂಟ್. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಮೂವ್ ಆನ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶನ್. So which statement is correct for bacterial transduction? So transduction is a type of sexual reproduction in bacteria where genetic material is exchanged between the two bacterial cells with the agency of virus. Okay. So transfer of genetic genes from one bacteria to another bacteria through virus. So this is the right option. Okay, got as transduction. Okay, so no need to read it again. So first option is correct. So transfer of gene from bacteria to another bacteria by conjugation. The type is called as conjugation itself. So bacteria obtain a DNA directly from mother cell. Yes. Okay. But it is not, of course the statement is correct, but it is not regarding the transduction. So bacteria obtain DNA from other external source. No, it's not. So this is the right answer. Exchange of genetic material between the two bacterial cells with the agency of virus is called as transduction. Okay. So question number 22. Yeah. So which of the is the following rank pair for the disease and its a causal organism? So smut of wheat, it is not caused by Puccinia. So smut of wheat caused by Eustylago medis. Okay. So that causes the smut of wheat. Eustylago, but Puccinia causes a rust of wheat. So it causes a rust of wheat caused by Puccinia. Okay. So smut of wheat. So white rust of crucifer caused by albigo. Early blight is caused by alternaria. Similarly, late blight is also there. Late blight. So which is the reason behind late blight is the reason behind Irish famine. Okay. So late blight caused by Phytophthora. Phytophthora infestans. It causes late blight. So early blight by alternaria. So early blight of potato. So potato-dalli ಎರಡು ತರ blight diseases ಗಳಿದೆ. Early blight by alternaria, late blight by Phytophthora. So red rot is caused by red rot in case of sugar cane. Red rot in sugar cane caused by Coleotrichum species. Okay, sugar cane. The species name is Coleotrichum fulcatum. Coleotrichum fulcatum. So there are different species of Coleotrichum over there. Where Coleotrichum fulcatum causes the red rot of sugar cane. Okay. So next question. Yeah. So sinusoidic mycelium is a characteristic of deuteromycete, ascomycete, mycete and basidiomycete. All these 1, 2 and 4 they have septate hyphae. Okay, whereas phycomycete have aseptate hyphae with multinucleate condition called as sinusoidic. So sinusoidic is a condition where fungi has aseptate hyphae with multinucleate condition, that is called sinusoidic. So phycomycete is the answer for this. So primitive fungi has that sinusoidic hyphae. Okay, whereas advanced fungi they have septate hyphae. Okay. Next question. So viroids have. So viroids have only genetic material without protein coat. Okay, so they have ssRNA enclosed in capsid, single stranded RNA, DNA enclosed in capsid, single stranded RNA without capsid. So they have single stranded RNA without capsid. So only genetic material is infectious here. Whereas in case of opposite, that is prions. So in case of prions, genetic material is absent but capsid is infectious. Okay, that is the difference between virus and prions. So viroids were discovered by Diner. Okay, potato spindle tuber disease. Okay, identified. Okay, so let's go to the next question. Yeah. So potato spindle tuber disease caused by virus particles still smaller than a virus. It is a nucleic acid. I already told it is a nucleoid. Only genetic material is infectious in case of So genetic material is infectious in case of viroids. Okay, so it is the answer. So only protein coat is infectious means it is a prion. Okay, so prion that causes some mad cow disease. Okay, so mad cow disease is caused by that prions. Okay, so both the protein coat and the nucleic acid, which is called as virus. ಎರಡು ಇದ್ರೆ ವೈರಸ್ಸು ಯಾವ್ದಾದ್ರು ಒಂದು ಲಾಸ್ ಇದ್ರೆ ಆ ವೈರಸ್ಸಲ್ಲಿ ಪ್ರೋಟೀನ್ ಕೋಟ್ ಲಾಸ್ ಇದ್ರೆ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಆಗುತ್ತೆ. ಸಾರಿ, ಓಕೆ, ಸೊ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಇರುವಂಥದ್ದು ವೈರಾಯ್ಡ್ ಆಗುತ್ತೆ. ಸೊ ಪ್ರೋಟೀನ್ ಕೋಟ್ ಮಾತ್ರ ಇದ್ದರೆ ಇಟ್ ಬಿಕಮ್ ಪ್ರಿಯಾನ್. ಓನ್ಲಿ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಇದ್ದರೆ ವೈರಾಯ್ಡ್ ಆಗುತ್ತೆ. ಓಕೆ, next question. Yeah, so this question number 26. Okay, So which is the correct sequence of the sexual cycle of fungi? So fungi ದಲ್ಲಿ ಫಸ್ಟ್ ಮೈಟಾಸಿಸ್ ಆಮೇಲೆ ಮಿಯಾಸಿಸ್ ಫರ್ಟಿಲೈಸೇಷನ್ ನೋ ಸಾಧ್ಯ ಇಲ್ಲ. ದೆನ್ ಪ್ಲಾಸ್ಮೋಗ್ಯಾಮಿ, ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ, ಮಿಯಾಸಿಸ್. So remember, plasmogamy occurs immediately in case of fungi during sexual reproduction, in case of ascomycete and basidiomycete. So plasmogamy occurs immediately, but it delays the karyogamy. After some time, karyogamy occurs, fusion of nucleus, undergo meiosis to produce haploid spores. Remember, fungi are haploids. So plasmogamy first agata, and immediately when the two hyphae are come to each other, ಇಲ್ಲಿರೋ ಕಂಟೆಂಟ್ ಎಲ್ಲ ಇಲ್ಲಿ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುತ್ತೆ. ಸೊ ಇಲ್ಲೊಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ, ದಿಸ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಆಲ್ಸೋ ಮೈಗ್ರೇಟೆಡ್ ದೇರ್. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ಕಂಟೆಂಟ್ ಖಾಲಿ ಆಗಿಬಿಡುತ್ತೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಫ್ಯೂಷನ್ ಆಫ್ ದ ಪ್ರೋಟೋಪ್ಲಾಸ್ಟ್ ಆಫ್ ದೀಸ್ ಟು ಈಸ್ ಬಿಕಮ್ ದ ಪ್ಲಾಸ್ಮೋಗ್ಯಾಮಿ. ಕ್ಯಾರಿಯೋಗ್ಯಾಮಿ ಇಮಿಡಿಯೇಟ್ ಆಗಿ ಆಗಲ್ಲ, ಇಟ್ ಡಿಲೇಸ್ ಫಾರ್ ಸಮ್ ಟೈಮ್, ದೆನ್ ಫ್ಯೂಷನ್ ಆಗುತ್ತೆ, ಮಿಯಾಸಿಸ್ ಆಗುತ್ತೆ. ಸೊ ಎರಡು ಡಿಫ್ರೆಂಟ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇರೋ ಸ್ಟೇಜ್ ಏನು ಬರುತ್ತೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಡೈಕ್ಯಾರಿಯಾನ್. ಓಕೆ, ಸೊ ಸೆಕೆಂಡ್ ಒನ್ ಈಸ್ ದ ರೈಟ್ ಆಪ್ಷನ್ ಫಾರ್ ದಿಸ್. Okay, yeah, next come to the next question. Yeah, so which of the following options is correct? So what is A, what is B, what is C? So A is the cell wall, B is the plasma membrane, C is nucleoid or genetic material, C is DNA, you can opt for that, cell membrane. Then D is heterocyst. Yeah, here is a H-shaped cell called a heterocyst, and E is a mucilage in a sheath. Yeah, so T second is the right answer. So here is the cell wall, cell membrane, genetic material, heterocyst, mucilage. Okay, so second is the right option. Yeah, next question. So which statement is not true about methanogens? So remember, methanogens are belongs to archaebacteria. They are archaebacteria. They live in marshy areas. That's right. Methane is their preferred carbon source. No. Okay. So they are present in guts of several ruminant animals. That's right. So methane is not preferred the carbon source. So that is not the right answer. So third is the correct for this option. Okay. Next one. The next question. Yeah. Protista includes, or protistans include heterotrophs, of course, chemo heterotrophs, of course they have photoautotrophs, yes they have, so all the above. Okay, so ella reethiya group of organisms, that's why we call protista is a mixed group. Ella ತರ organisms ಆ group ನಲ್ಲಿ ಇರುತ್ತೆ, that's why they are mixed organisms. There is no definite boundary for the describing a character. Okay, so bacteria are found to be primitive organisms because ಯಾಕೆ ಅದನ್ನು ತುಂಬ ಪ್ರಿಮಿಟಿವ್ ಅಂತ ಹೇಳ್ತೀವಿ? ದೇ ಆರ್ ಸ್ಮಾಲ್ ಮೈಕ್ರೋಸ್ಕೋಪಿಕ್ ವಿಚ್ ಆರ್ ನಾಟ್ ಸೀನ್ ವಿತ್ ನೇಕೆಡ್ ಐ. ಇದೇ ಥರ ಬೇಕಾದಷ್ಟು ಆರ್ಗ್ಯಾನಿಸಮ್ ಇದೆ. ದಟ್ ಈಸ್ ನಾಟ್ ದ ರೀಸನ್ ಕಾಸ್ ಸೀರಿಯಸ್ ಡಿಸೀಸಸ್. ಸೊ ಡಿಸೀಸ್ ಕಾಸ್ ಮಾಡ್ತದೆ ಅಂದ ತಕ್ಷಣ ಇಟ್ ಇಸ್ ನಾಟ್ ಕನ್ಸಿಡರ್ಡ್ ಆಸ್ ಎ ಪ್ರಿಮಿಟಿವ್. ಪ್ರೊಡ್ಯೂಸಸ್ ಎಂಡೋಸ್ಪೋರ್ ವಿಚ್ ಆರ್ ವೆರಿ ರೆಸಿಸ್ಟೆಂಟ್ ಟು ಅಡ್ವರ್ಸ್ ಕಂಡೀಷನ್. ಸೊ ಎಂಡೋಸ್ಪೋರ್ ಆದ ತಕ್ಷಣವೇ ಇಟ್ ಇಸ್ ನಾಟ್ ಕನ್ಸಿಡರ್ಡ್ ಆಸ್ ಎ ಪ್ರಿಮಿಟಿವ್. ಸೊ ಪೊಸೆಸ್ ನ್ಯೂಕ್ಲಿಯೈಡ್. ಯಾ, ದೇರ್ ಇಸ್ ನೋ ಡೆಫಿನೆಟ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಆ್ಯಂಡ್ ಶೋ ಎ ಮೈಟಾಟಿಕ್ ಡಿವಿಷನ್. ಸೊ ಎ ಮೈಟಾಟಿಕ್ ಡಿವಿಷನ್ ಇಟ್ ಇಸ್ ನಾಟ್ ಎ ಟ್ರೂ ಮೈಟಾಟಿಕ್, ಟ್ರೂ ಡಿವಿಷನ್ ಅಲ್ಲ ಅದು, ಹಾಗಾಗಿ ಅದನ್ನ ಎ ಮೈಟಾಟಿಕ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಆಪ್ಷನ್ 4. ಓಕೆ, the next question. Okay, so which option is correct for a structure of a cell wall of a fungi and bacteria? So ಎರಡಕ್ಕೂ same conditions ಇದೆ. Both have N-acetylmuramic acid. No, bacteria have N-acetylmuramic acid, but in fungi it is absent. Both are made up of N-acetylglucosamine. Okay, okay, so glucosamine amine groups fungi nallu irutte, so chitin nalli irutte. Then both are made up of N-acetylglucosamine and acetylmuramic acid. Idu fungi bacteria maatra agutte. Both are made up of chitin. No, so right option is second. So amino group fungi nallu irutte, bacteria nallu irutte. That is a common thing, N-acetylglucosamine. So that is called NAG, NAG, NAG. N-acetylmuramic acid is NAM. So NAM and NAG are present in bacteria, whereas amino group is present in. Okay, so by considering all this we can choose B. Third is the right option. Okay, so let's move on to the next question. Question number 34. Okay, so which of the following combinations of characteristics are true for slime molds? So ಯಾವ slime molds ಗೆ true characters? Okay. So parasitic plasmodium with the true walls, spore dispersed by air current. Of course, spores are dispersed by air current, but they are not parasitic. Saprophytic plasmodium with walls. So wall is absent in plasmodium. So naked protoplast irutte. So it is called plasmodium. Parasitic. So parasitic is no. Saprophytic yes. Plasmodium without wall yes. Spores dispersed by air current, and spores they have cell wall. So spore cell matra cell wall irutte. So that is a unique character of slime molds. So slime molds comes under saprophytic protozoans. Okay. So autotrophic protozoans are dinoflagellates and chrysophytes. They are photosynthetic protozoans. Okay. Next question number 35. 35. So match to the following. So phytoplanktons. So phytoplanktons andre the smallest plants which are float in the water called as phytoplanktons. Animals like it are zooplankton. So phytoplanktons andre diatoms. So dinoflagellate example Gonulax, Mixotropes example Euglena, Protozoan example Paramecium. Okay. So nodkoli A4. So A4 erediದೆ. So you can eliminate them. Then B, dinoflagellate 3, B3, so you can choose this. So this is the right answer. Okay. So then C2, Mixotrope, Paramecium. Yeah, that's right. And come to the next question. Okay. So homothalism means fusion of two gametes from two thalli of opposite strands, fusion of two gametes from thalli of a similar strand. So homo means the same kind. ಒಂದೇ ಐ ಫಂಗಲ್ ಐಫೆ ಮೇಲೆ ಎರಡೂ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಬೆಳೀತಾ ಇದ್ರೆ ಇಟ್ಸ್ ಕಾಲ್ಡ್ ಹೋಮೋಥ್ಯಾಲಿಕ್. ಬೇರೆ ಬೇರೆ ಫಂಗಸ್ ಐಫೆಯಿಂದ ಬರ್ತಾ ಇದ್ರೆ ದೇ ಆರ್ ಕಾಲ್ಡ್ ಎಟಿರೋಥ್ಯಾಲಿಕ್. ಸೊ ಒಂದೇ ಫಂಗೈ ಮೇಲೆ ಬೆಳೀತಾ ಇದೆ. ಸೊ ಇಟ್ ಈಸ್ ಫ್ಯೂಷನ್ ಆಫ್ ಟೂ ಗ್ಯಾಮೆಟ್ಸ್ ಫ್ರಮ್ ಥ್ಯಾಲೈ ಆಫ್ ಅ ಸಿಮಿಲರ್ ಸ್ಟ್ರಾಂಡ್. So then fusion of two gametes of a same thallus, fusion of two gametes of produced from the same thallus. So both 2 and 3 are the right option for this. Okay. So homothallic. If they are from two different, it is called as heterothallic. ಎರಡು ಕಡೆ ಬೇರೆ ಕಡೆಯಿಂದ ಬರ್ತಾ ಇದ್ರೆ it is called as heterothallic. Next come to the question number 37. So peptidoglycan is a characteristic constituent of the cell wall of peptidoglycan ಯಾವುದರಲ್ಲಿ ಇರುತ್ತೆ? ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಮತ್ತು ಯೂನಿಸೆಲ್ಯುಲರ್ ಯೂಕ್ಯಾರಿಯೋಟ್ಸ್. ಸೊ ಯೂಕ್ಯಾರಿಯೋಟ್ಸಲ್ಲಿ ಇರಲ್ಲ. ದೆನ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆ್ಯಂಡ್ ಸೈನೋಬ್ಯಾಕ್ಟೀರಿಯಾ. ಆರ್ಕಿಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಸೂಡೋಮ್ಯೂರಿನ್ ಇರುತ್ತೆ. ಆಲ್ ದ ಮೆಂಬರ್ಸ್. ಓಕೆ. ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆ್ಯಂಡ್ ಸೈನೋಬ್ಯಾಕ್ಟೀರಿಯಾ ಈಸ್ ದ ಆನ್ಸರ್. ಓಕೆ. ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೈನೋಬ್ಯಾಕ್ಟೀರಿಯಾ ಎರಡರಲ್ಲೂ ಕೂಡ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್ ಆರ್ ಮ್ಯೂರಿನ್ ಇರುತ್ತೆ. ಆರ್ಕಿಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಸೂಡೋಮ್ಯೂರಿನ್ ಇರುತ್ತೆ. ಯೂಕ್ಯಾರಿಯೋಟ್ಸಲ್ಲಿ ಡಿಫ್ರೆಂಟ್ ಇರುತ್ತೆ. ಓಕೆ. ಸೊ ಆಲ್ ಮೆಂಬರ್ಸ್ ಆಫ್ ಮೊನೇರಾ ದೇ ಡೋಂಟ್ ಹ್ಯಾವ್ ದ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್. ಯಾಕೆಂದರೆ ಆರ್ಕಿಬ್ಯಾಕ್ಟೀರಿಯಾ ಮೊನೇರಾದಲ್ಲೇ ಬರೋ ಆರ್ಕಿಬ್ಯಾಕ್ಟೀರಿಯಾ ದೇ ಹ್ಯಾವ್ ಸೂಡೋಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್. ಓಕೆ. ಸೊ ರೈಟ್ ಆಪ್ಷನ್ ಈಸ್ ಸೆಕೆಂಡ್. ಓಕೆ. ನೆಕ್ಸ್ಟ್ ಕ್ವೆಶ್ಚನ್ ನಂಬರ್ ಹೆಟಿರೋಸಿಸ್ಟ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ BGA ಬ್ಲೂ ಗ್ರೀನ್ ಅಲ್ಗೆ ಅಂತ ನಾಸ್ಟಾಕ್ನಲ್ಲಿ ಪಾರ್ಟಿಸಿಪೇಟ್ ಇನ್. ಸೊ ಹೆಟಿರೋಸಿಸ್ಟ್ ಫಂಕ್ಷನ್ ಏನು? N2 ಫಿಕ್ಸೇಷನ್. ಸೊ ಇನ್ವಾರ್ಡ್ ಇನ್ ನೈಟ್ರೋಜನ್ ಫಿಕ್ಸೇಷನ್. Participate in it, and they lack what? So PS2 and PS1, you might know that what is PS1 and PS2. So PS1 and PS2 are photosystem 2 and photosystem 1. In blue green algae, the heterocyst they have nitrogen fixation ability and they lack PS2. PS2 is absent there. Okay. So only PS1 is there. Okay. So if you remember the functions of PS2 and PS1, so PS2 only if PS2 is involved and lack PS2. Okay. So PS2 iralla. So PS2 illa antandre photolysis of water agalla. Okay. That is the thing there. Okay. So 30 height aitu. Okay. Next question number 39. The sequence of 16S ribosomal RNA genes was used in differentiating. So 16S rRNA, it is used to differentiate archaebacteria from eubacteria. So by Carlus differentiate archaebacteria and eubacteria based on the sequence of 16S rRNA. Okay, so remember this important. Advanced character na consider ಮಾಡ್ಕೊಂಡು he classify or split Monera into archaebacteria and eubacteria. Okay, so 39. Next, last question. So regarding the following figure, identify the correct answer. A ಯಾವುದು, B ಯಾವುದು, C ಯಾವುದು ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೊ A mucor, B Rhizopus or Aspergillus, C ಯಾವುದು ಮಶ್ರೂಮ್. ಅಫ್ಕೋರ್ಸ್ ಮಶ್ರೂಮ್ಸ್ ಅಗಾರಿಕಸ್ ಎರಡೂ ಕೂಡ ಸೇಮ್ ಆಗುತ್ತೆ. ಅಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಆನ್ಸರ್ ದೇ ಆರ್ ಎಕ್ಸ್ಪೆಕ್ಟಿಂಗ್. ಓಕೆ. ಸೊ ಸಿ ಯಾವ್ಯಾವ ಚಾನ್ಸಸ್ ಇರುತ್ತೆ ನೋಡಿ, ಮಶ್ರೂಮ್ ಹೌದು, ಅಗಾರಿಕಸ್ ಹೌದು, ಎಲ್ಲದೂ ಆಗುತ್ತೆ. ಬಿ ರೈಸೋಪಸ್ ಆರ್ ಆಸ್ಪರ್ಜಿಲಸ್ ಆರ್ ಪೆನ್ಸಿಲಿಯಂ ಆರ್ ಆಸ್ಪರ್ಜಿಲಸ್. ಎ ಮ್ಯೂಕರ್. ಸೊ ಆ್ಯಕ್ಚುಲಿ ಇಟ್ ಈಸ್ ಮ್ಯೂಕರ್ ಆಸ್ಪರ್ಜಿಲಸ್. ಸೊ ಮ್ಯೂಕರ್ not Rhizopus. Yeah, ಇಲ್ಲಿದೆ ನೋಡಿ, ಮ್ಯೂಕರ್, ದೆನ್ ಆಸ್ಪರ್ಜಿಲಸ್, ದೆನ್ ಅಗ್ಯಾರಿಕಸ್. Okay, so this is the right answer. So it completes the chapter. Thank you.